Let's learn about macromolecules or biomolecules. Living organisms exhibit astonishing diversity in their form, structure, behavior, and mode of life, yet their chemical makeup, molecular architecture, and metabolic pathways are strikingly similar. All of them are made up of the same chemical elements and similar organic molecules, and all their life processes take place in a medium of water. The fact that all organisms share the same biochemical composition provides a strong evidence in support of their evolutionary kinship. Living matter is a uniquely complex and mysterious arrangement of atoms, ions, molecules, and colloidal particles. Its constituent molecules are generally called biological molecules or simply biomolecules. Biologically important elements. The elemental composition of the living matter is very different from that of atmosphere and lithosphere. All about 22 out of 100 odd elements of the periodic table are essentially found in living organisms. They are mostly light elements and are generally called biogenic elements. Nearly 16 of them are found in all groups of organisms. The distribution of these elements in living organisms is not in proportion to their occurrence in Earth's crust. The most abundant ones among them are in the order H, O, N, and C. The other important biogenic elements include Na, K, Ca, Mg, P, S, Cl, and Fe. Of all the biogenic elements, C, H, O, and N are universally present, and they account for nearly 99% of the mass and number of the atoms in the living matter. These four elements have some common features. Of all the biogenic elements, C, H, O, and N are universally present and they account for nearly 99% of the mass and number of the atoms in the living matter. These four elements have some common features. They are light and they can form fairly strong covalent bonds with each other and also with other elements by sharing electrons. Their biological predominance is largely due to their ability to form stable covalent bonds. These four elements have some common features. They are light and they can form fairly strong covalent bonds with each other and also with other elements by sharing electrons. Their biological predominance is largely due to their ability to form stable covalent bonds. H, O, N, and C are the lightest biogenic elements capable of forming 1, 2, 3, or 4 covalent bonds by attracting 1, 2, 3, or 4 electrons respectively to complete their outer electron shells. Since the strength of the covalent bond is inversely proportional to the atomic weight of the bonded atoms, these elements are biologically best suited for covalent bonding. C, O, and N can form double or triple bonds, which can greatly increase the variety of molecular configurations. The compounds of carbon found in living organisms are highly reduced or hydrogenated. The reduced organic molecules found in living matter are energy rich, since their synthesis is an energy consuming and energy storing endergonic process, and living organisms spend free energy for it. Fitness of carbon for life. Carbon is perhaps the most important biogenic element. It's unique among all elements for the number and variety of the compounds it can form. More than a quarter million carbon compounds have already been identified and described. Since carbon-containing molecules are almost exclusively associated with living matter, they form the chemical basis of life. The significant features of carbon, uniquely suited to its biological role, are the following. Carbon is a light element with the atomic number 6. Its atoms are relatively small and light with low mass. Carbon is the only element which possesses both slow reactivity and infinite structural versatility. Most reactions of carbon compounds are slow, even though some of them may involve the release of a great deal of energy. A carbon atom has 4 valencies, so it can form 4 covalent single bonds or 2 double bonds either with other carbon atoms or with the other kinds of atoms such as H, O, N, and S and halogens. These carbon atoms can join together and form chains or rings of any size, sharing 2 electrons between each pair. This ability of carbon or any other element to form covalent bonds between its own atoms for the formation of rings or chains of infinite length is known as the power of catenation. Catenation gives carbon great potential for the formation of carbon skeletons or carbon frameworks, a great variety of atomic combinations, and an infinite variety of molecular structures. So, which one was the first point? Carbon is a light element with the atomic number 6. Its atoms are relatively small and light with low mass. Carbon is the only element which possesses both slow reactivity and infinite structural versatility. Okay, so covalent bonding between carbon atoms can be seen, and carbon chain is an octane, carbon ring is a benzene, double bond, triple bond, covalent carbon bonding can be seen. Covalent bonding between carbon atoms forms stable rings, straight and linear, or branching chains and complex networks, which all serve as the backbone of an endless variety of organic molecules. Attached to this carbon skeleton are groups of other atoms called functional groups, which confer specific properties on the molecules. Covalent bonding between carbon atoms forms stable rings, straight and linear or branching chains, and complex networks, which all serve as the backbone of an endless variety of organic molecules. Attached to this carbon skeletons are groups of other atoms called functional groups, which confer specific properties on the molecules. In organic molecules, carbon atoms can bond in more than one arrangement. In organic molecules, carbon atoms can bond in more than one arrangement, giving rise to different compounds with different structures. This phenomenon, in which compounds have the same molecular formula but different arrangement of atoms, is termed isomerism. Multiple covalent bonding of carbon atoms with hydrogen, oxygen, nitrogen, sulfur, and halogens forms an at most infinite variety of functional groups and molecular structures and configurations. Carbon has the ability to react with both electropositive elements such as oxygen, nitrogen, phosphorus, sulfur, and chloride, and electronegative elements, hydrogen. Multiple covalent bonding of carbon atoms with hydrogen, oxygen, nitrogen, sulfur, and halogens forms an almost infinite variety of functional groups and molecular structures and configurations. Carbon compounds are stable and inert except in the presence of catalysts. This is because a saturated carbon compound is neither a donor nor an acceptor of electrons or atoms. Carbon compounds are stable and inert except in the presence of catalysts. This is because a saturated carbon compound is neither a donor nor an acceptor of atoms. So let's learn about the classification of biogenic elements. Biogenic elements fall under four major categories, namely macrobiogenic, oligobiogenic, microbiogenic, and ultramicrobiogenic. Macrobiogenic elements or major elements are those elements commonly found in all organisms in high proportions. They include CH, O, and N. Oligobiogenic elements are the elements occurring less frequently in small proportions. They include Na, K, Ca, Mg, S, P, Fe, etc. Some of them are the constituents of some biologically important molecules. For example, P is an essential constituent of ADP, nucleic acids, nucleotides, etc. Mg is an essential constituent of chlorophyll, and Fe is an essential constituent of hemoglobin and cytochromes. Na and K are essential for the cells that are involved in the transmission of impulses, and Ca is essential for muscular contraction. Magnesium is an essential constituent of chlorophyll, and iron is an essential constituent of hemoglobin and myoglobin, along with cytochromes. Na and K are essential for the cells that are involved in the transmission of impulses. Ca is an essential for muscular contraction, hemoglobin, cytochromes. Microbiogenic elements are the elements which occur in very small amounts. They include Cu, Co, Mn, Zn, Cl, F, Bri, etc. Some of them serve as cofactors in enzyme actions. Ultra microbiogenic elements are the elements found in negligible or trace amounts. They include PO, AL, NI, CD, CRAS, SI, SE, TI, GA, BI, etc. Molecular basis of the living matter. The constituent elements of the living matter exist mostly as compounds. The constituent elements. The constituent elements. The constituent elements of the living matter exist mostly as compounds. Inert gases, nitrogen and oxygen may be found in molecular form, dissolved in body fluids. These compounds may be ionic or molecular and organic or inorganic. The inorganic compounds include water and mineral salts. Mineral salts occur either as anions or as cations. Organic biomolecules are significant in that they largely determine the organizational features and functional properties of living matter. Molecular basis of living matter. The constituent elements of the living matter exist mostly in compounds. The constituent elements of the living matter exist mostly as compounds. Inert gases, nitrogen and oxygen may be found in molecular form, dissolved in body fluids. These compounds may be ionic or molecular and organic or inorganic. These inorganic compounds include water and mineral salts. Mineral salts occur either as anions or as cations. Organic biomolecules are significant in that they largely determine the organizational features and functional properties of the living matter. They include carbohydrates, proteins, lipids, enzymes, vitamins, and hormones. Most organic biomolecules are derivatives of hydrocarbons. These hydrocarbons are compounds with a stable and covalently linked carbon backbone to which hydrogen atoms are bonded. In some places, hydrogen atoms may be replaced by different functional groups, yielding different families of organic compounds. Hydroxyl, carbonyl, aldehyde and keto, methyl phenyl ether ester amino amido, guanidino, imidazole, sulfhydryl, and disulfide groups are the common functional groups. Alcohols, amines, aldehydes, ketones, and carboxylic acids are the typical families of organic compounds. So, Na and K are involved in the transmission of impulses, and calcium is associated with the muscular contraction. Micromolecules and macromolecules. In general, organic molecules are of two major groups, namely micromolecules and macromolecules. Micromolecules are small, low molecular weight substances. Some of them are monomers formed of a single unit, and the others are oligomers formed by a few units. Simple sugars, amino acids, fatty acids, nucleotides, etc. are micromolecules. So protoplasmic compounds are of two types, namely organic and inorganic type. Micromolecules are simple sugars, mono and oligosaccharides, amino acids, fatty acids, glycerol, nucleotides, and macromolecules are of polysaccharides, peptides and proteins, fats, nucleic acids, enzymes, hormones, vitamins, inorganic type, water and mineral salts. Molecular constituents of protoplasm can be classified into two types, mainly by the organic and inorganic type, which in turn the organic part can again be transformed into two types like the micromolecules and macromolecules. Macromolecules, simple sugars, amino acids, fatty acids, glycerol, nucleotides. Macromolecules, polysaccharides, peptides and proteins, fats, nucleic acids, enzymes, hormones and vitamins. So macromolecules are giant molecules with large size and high molecular weight. The principal organic macromolecules of the living matter include proteins, polysaccharides, lipids, nucleic acids, enzymes, hormones and vitamins. A macromolecule in turn is a complex chain of polymer of several repeating monomeric units linked through covalent bonds. If a polymer is formed of one and the same kind of monomeric units, it is called a homopolymer. If it is formed of different kinds of monomers, it is called heteropolymers. Glycogen is a homopolymer formed of only glucose units. Glycogen is a homopolymer formed of only glucose units. Fats or triglycerides are heteropolymers formed of fatty acid and glycerol. So glucose plus glucose, infinite times glucose, leads to glycogen, which is a homopolymer. And if fatty acids and glycerols are added together, then it creates fats to form heteropolymers. Now we are going to learn about the asymmetry of biomolecules. Asymmetry of biomolecules is a molecular structure in which an atom having 4 tetrahedral valencies is linked to 4 different atoms or functional groups in a tetrahedral arrangement. Such an asymmetric atom is called chiral atom or chiral center. In most cases, the asymmetric atom is carbon, but in some cases, nitrogen or sulfur may become asymmetric. A carbon atom is said to be asymmetric when it bears 4 different atoms or groups of atoms at its 4 valency bonds. In glyceraldehyde, lactic acid, amino acids, etc., there is only a single asymmetric carbon. But in sugars, tartaric acid, terpenes, etc., more than one asymmetric carbon atoms are present. The 4 different groups attached to the asymmetric carbon of glyceraldehyde are CHO, aldehyde, HOH, hydroxyl, and CH2OH, and those attached to the asymmetric carbon of lactic acid include CH3, methyl, COOH, carboxyl, H, and OH. So glyceraldehyde can be seen where carbon is attached to another carbon along with an oxygen with double bonds and a hydrogen is there. Then the middle carbon has one H bond and then an OH linkage along with a CH2H linkage. So the central one has 4 different groups. Tetrahedral valencies are linked to 4 different atoms or functional groups like HOH, CH2H, COO, all like that. The asymmetric molecules that are mirror images of each other are called chiral molecules or enantiomers. They are related to each other optically at right and left hands and are characterized by optical activity. So isomerism of biomolecules. Isomerism is the existence of two or more chemical compounds with the same molecular formula but with different structural formulae or different spatial arrangement of atoms. In other words, it is the molecular organization in which different molecules have the same number and kind of atoms and the same molecular weight and molecular formula but have different arrangement or configuration of constituent atoms. The molecules which exhibit isomerism are called isomers. For example, butyl alcohol and ethyl ether have the same empirical formula C4H10O, but they are entirely different kinds of substances with different molecular configuration or atomic arrangements. Isomerism of biomolecules is of 2 major kinds, namely structural or constitutional isomerism and stereoisomerism or space isomerism, structural one and stereoisomerism. So first learn about the structural isomerism. Structural isomerism is the condition in which the isomers have the same molecular formula but have the different structures. So they differ from each other in the order in which their atoms are bound together. So there are different kinds of structural isomerism. There are different types of structural isomerism such as chain isomerism, position isomerism, functional isomerism, metamerism, and tautomerism. So chain isomerism, and we are going to learn in the next session, chain isomerism. This is the structural isomerism in which the isomers differ from each other in arrangement of the carbon chain within their molecules. Chain isomerism, this is the structural isomerism in which the isomers differ from each other in the arrangement of the carbon chain within their molecules. Butane and isobutane, this molecular formula is C4H10. This can be arranged in different types of chains. So next is the position isomerism. The isomerism in which the isomers have the same carbon chain but they are different in the position of structural entity such as a multiple bond, functional group, etc. 1-butene and 2-butene are position isomers with the molecular formula C4H8. Functional isomers can be different. This is the isomerism in which isomers differ from each other with respect to their functional groups Ethanol and dimethyl ether are functional isomers with the molecular formula C2H6O. Next category is the metamerism one. The structural isomerism in which the isomers or metamers belong to the same homologous series but differ in the number of the alkyl groups attached to the two sides of the bivalent functional group. Example, ethoxymethane and methoxymethane. Tautomerism. Tautomerism is the structural isomerism in which the two tautomers are interconvertible and they can transform to each other. So they are in dynamic equilibrium with each other. Thus, interconversion of tautomerism is due to the intramolecular shift of migration of an atom or a group from one location to another. Tautomers differ from each other in the locations of atoms and electrons. In most cases, tautomerism results from the migration of the hydrogen atom and the change in the location of the double bond. Such a type of tautomerism is called proton tautomerism, and the tautomers involved are called proton tautomers, etc. Acetaldehyde and vinyl alcohol. Acetaldehyde and vinyl alcohol can be seen. This is an example of keto-enol transformation of tautomerism, in which a keto tautomer, acetaldehyde, tautomerism is a structural isomerism in which the two isomers or tautomers are interconvertible and they can transform to each other. So they are in dynamic equilibrium with each other. This interconversion of tautomers is due to an intermolecular shift of migration of an atom or a group from one location to another. Tautomers differ from each other in the locations of atoms and electrons. In most cases, tautomerism results from the migration of a hydrogen atom and a change in the location of a double bond. Such a type of tautomerism is called a proton tautomerism, and the tautomers involved are called proton tautomers. Acetaldehyde and vinyl alcohol. Acetaldehyde a keto form, vinyl alcohol is an enol form. This is an example of keto-enol tautomerism in which a keto tautomer, acetaldehyde that contains a carbonyl keto group, exists in equilibrium with an enol tautomer, vinyl alcohol, that contains a hydroxyl enol group. Next is the stereoisomerism. Stereoisomerism or space isomerism is the condition in which the isomers have the same number of atoms, the same kinds of groups and the same molecular formula and the same structure, but have different configurations in space. In other words, it is the molecular organization in which different molecules have the same structure but different spatial arrangement of atoms or groups. Thus, stereoisomers are the compounds or molecules which have identical chemical composition but have different arrangement of atoms or groups in space. Stereoisomerism is in fact the spatial arrangement of an organic molecule either due to the presence of double bonds around which there is no freedom of rotation or due to the presence of chiral centers around which constituent groups are arranged in definite order. Since the isomers differ from each other in the arrangement of other atoms in space, the arrangement is called stereoisomerism or space isomerism. There are different kinds of stereoisomerism. Stereoisomerism is of two main kinds: conformational isomerism and configurational isomerism. In the former, or the conformational isomerism, the isomers differ from each other in their conformation due to differences in the spatial arrangement of their atoms or groups. Conformation is a particular shape of a molecule that results from the normal rotation of its atoms or groups about single bonds. This conformational isomerism arises due to the free rotation of atoms or groups about single bonds. In configurational isomerism, the isomers differ from each other in their configuration. Configuration refers to the specific spatial arrangement of atoms or groups within a molecule. Configurational isomerism is of two kinds, namely geometrical or cis-trans isomerism and optical isomerism. So what is geometrical or cis-trans isomerism? Geometrical isomerism, currently termed cis-trans isomerism, is a type of stereoisomerism in which the isomers have the same structural formula but have different spatial arrangement of atoms or groups around a non-rotating double bond. It results from the restricted rotation about a double bond between two atoms. Thus, geometrical isomerism is the stereoisomerism due to restricted rotation about double bonds. The mere presence of a double bond in a molecule by no means ensure the occurrence of geometrical isomerism. The most essential condition or prerequisite for geometrical isomerism to occur is that two identical atoms or groups should not be present on the two atoms linked by a double bond. For example, molecules like ABC double bond CAB or ABC double bond CED, etc. exhibit geometrical isomerism, whereas AAC double bond CAA, AAC double bond CB, AB, etc. do not exhibit geometrical isomerism. A geometrical isomer with similar groups on the same side of the double bond is called cis isomer, and the isomer with similar groups on the opposite sides of the double bond is called trans isomer. So, geometrical isomerism is more aptly termed cis-trans isomerism. In cis-trans geometrical isomerism, one isomer will exhibit cis arrangement and the other one will exhibit trans arrangement. The cis-trans pair fumaric acid and maleic acid with the molecular formula C4H4O4 is an example. Next is the optical isomerism. This is a type of stereoisomerism in which the isomers resemble each other in most of their properties but differ from each other in their optical activity or behavior towards plane polarized light. In it, a molecule can exist in left-handed and right-handed forms which are mirror images of each other. Optical isomerism is related to the optical activity of biomolecules. Optical activity is the ability of a molecule to rotate to the plane of polarization of a plane polarized light, either to the left or to the right. It is exhibited only by those molecules which have one or more asymmetric or chiral carbon atoms. Molecules without asymmetric carbon atom cannot rotate plane polarized light. The degree of rotation depends on the concentration of the compound. In optical isomerism, an asymmetric chiral molecule exists in two optically active forms called optical isomers. These optical isomers are optically active stereoisomers. The optical isomers of a molecule have the same chemical composition but have different spatial configurations. The number of the optical isomers of a molecule depends on the number of its asymmetric atoms. It is always equal to 2 raised to n, where n represents the number of asymmetric atoms. Thus, a molecule having two asymmetric atoms will have 4 optical isomers, like 2 raised to 2. Optical isomers are capable of optical rotation. Optical rotation is the change of direction of the plane polarized light either to the right or to the left as it passes through a molecule containing one or more asymmetric carbon atoms. Example, sugars. The two mirror images forms of an asymmetric molecule are optical isomers. One of them will rotate polarized light to the right and the other one to the left at the same degree. The rotation of a plane polarized light towards the right is called dextrorotation and towards the left is called levorotation. Levorotation. Dextrorotation could be clockwise and levorotation could be anti-clockwise to an observer viewing the light coming towards him. Dextrorotation is indicated either by a plus sign or by an italic D. Similarly, levorotatory is indicated either by a minus sign or by an italic L. Thus, it becomes clear that optical isomers are either of two types, namely dextrorotatory isomers, D isomers or D+, and levorotatory isomers, L isomers or L-. Usually, molecules having right-handed configuration, D isomers, are dextrorotatory, and those having left-handed configuration, L isomers, are levorotatory. A mixture of D and L isomers in equal amounts will not show optical activity. Such a mixture is sometimes called DL form, plus or minus form, racemic mixture or racemate. So a plane polarized light is rotated counterclockwise, which shows a levorotation, and plane polarized light before rotation is straight, but after plane polarized light rotates clockwise. That's the phenomenon of dextrorotation. Lactic acid is a typical example of D and L isomers. 3 forms of it can be recognized, namely dextro, levo, and racemic forms. Dextrolactic acid is a product of muscle metabolism and hence it is also called sarcolactic acid. Sarcolactic acid. Levolactic acid is a product of the action of some microorganisms on lactose. Laboratory synthesis of lactic acid produces an inactive and equimolar mixture of D and L forms, often called racemic lactic acid, plus or D lactic acid, minus or L lactic acid. Enantiomers and diastereomers. Optical isomers are of two kinds: enantiomers, and diastereomers, diastereoisomers. Enantiomers are non-superimposable mirror images of each other, whereas the diastereomers are not mirror images. This can be illustrated as follows: L isomer, D isomer, L isomer, and D isomer. In the above example, 1 and 2 represent a pair of enantiomers and so are 3 and 4, but 1 and 3 and 2 and 4 are diastereomeric pairs. Enantiomers are a pair of optical isomers whose molecular configurations have left-handed and right-handed chiral forms. These forms are conventionally called dextro and levo forms because they are structural mirror images of each other, just as our right and left hands. The notations D and L have no bearing with the rotation of polarized light. They represent the configuration of the two enantiomers or mirror images of a molecule. The notations D or plus and L minus or minus denote the rotation of the plane polarized light. So they represent the dextrorotatory and levorotatory forms respectively. Thus D-glucose is dextrorotatory D, whereas D-fructose is levorotatory. Equimolar mixtures of optical isomers will not have net rotation, so they are called racemic mixtures. Enantiomers are usually found in compounds with a single asymmetric carbon atom. The enantiomeric forms of glyceraldehyde and those of 2-butanol are examples. These are mirror images of each other and cannot be made to coincide. Enantiomers of glyceraldehyde, enantiomers of 2-butanol can be seen. Diastereoisomers are a group of 4 optical isomers occurred in compounds with 2 asymmetric carbon atoms such as 4-carbon sugars, and 4 isomers include 2 pairs of enantiomers, namely a pair of D forms and a pair of L forms. The 2 D isomers are mirror images of each other and so are the 2 L isomers. However, the 2 D forms are not mirror images of the L forms or vice versa, so they are called diastereoisomers. Since compounds with asymmetric carbon atoms can exist in half right-handed and left-handed isomeric forms, they are called chiral compounds, and their asymmetric carbon atom is termed chiral atom or chiral center. Amino acids, with the exception of glycine, glucose, etc., are chiral molecules. Amino acid has a single chiral center and glucose has 5. In living organisms, chiral molecules used to exist only in one of their chiral forms. For example, in natural proteins, amino acids occur only as L-amino acids, and in carbohydrates, glucose occur only as D-glucose.